to Dr. Patrick Forcelli for this conference. Dr. Patrick Forcelli is a Ph.D. professor and chair of the Department of Pharmacology and Physiology at Jerome H. Fleisch and Marlene L. Cohen Endowed. He is professor of pharmacology and also director of graduate students Ph.D. in pharmacology and physiology at Georgetown University Medical Center in Washington. I appreciate a lot this conference because it's very important for us to talk about different topics associated with epilepsy. Please go ahead, Patrick.
Thank you so much, Louisa, for the introduction. It's really a pleasure to be able to tell you about some of the work that my lab has been doing over the last few years, focused around cellular senescence as a target to treat the epilepsies. And I want to start off by acknowledging the grant funding that supported this work, so NIH grants and a Department of Defense grant, as well as two PhD students, Ty Kahn and David McFall, who did a lot of this work, and a postdoc in my lab, Max Stevenson, who has also contributed quite a bit. So I don't have to belabor this point. The current therapies that we have to treat the epilepsies are symptomatic treatments. Despite the development of many, many anti-seizure medications over the last 100+ years, and you can see the generation here, none of these drugs are truly disease modifying, with perhaps the exception of everolimus in tuberous sclerosis complex. So as a result, we have lots of medications that can control seizures, but none that can prevent the development of epilepsy after injury, and none that modify epilepsy once it occurs. This is an important area for both preclinical and clinical development in the epilepsies, disease-modifying therapies. Fortunately, we have multiple places where we can intervene. This is where our current anti-seizure medications are working. After the emergence of epilepsy and spontaneous seizures, individuals have recurrent seizures. Fortunately, 2/3 are going to experience good seizure control on our current anti-seizure medications. Of course, we have about 1/3 who are going to have recurrent or drug refractory seizures. But by this point, the epilepsy is already developed. So we're looking towards earlier stages, the latent period after an injury, as an opportunity and a window for disease-modifying therapies. So, we can think about this as a series of phases. We have an acute injury phase. This could be traumatic brain injury, stroke, infection, or a common model, status epilepticus. This leads to secondary injury, including things like DNA damage, apoptosis, upregulation of pro-inflammatory cytokines, oxidative stress. Many of which persist during the latent period, the period of reorganization between initial injury and the development of chronic epilepsy, spontaneous seizures, and emergence of cognitive deficits. So in humans, we're talking about timescales of minutes to hours for the acute injury, months to years for the latent period, and years to decades for chronic epilepsy. In rodents, again, minutes to hours for our acute injury, days to weeks to maybe months for the latent period, and then months to years for the chronic epilepsy. So that's just by way of orienting us to what our goal is, how we can intervene. The other piece that I'd like to tackle by way of background is this concept of cellular senescence. So these are two figures from papers by Hayflick in 1961 and 1965. And many of you have heard of the Hayflick limit. So what he observed was culturing mammalian cells in vitro rather. He found that with repeated cell division, there would be a phase in which the primary culture would grow, and then another phase where growth would start to slow down, and then a third phase where there would be a loss in cells. When he quantified this as number of serial passages as they split these cells, he found again that there was a phase in which they were, dividing pretty stably, and then a phase at which divisions ceased. He said in this paper, consequently, we advanced the hypothesis that the finite lifetime of diploid cell strains in vitro may be an expression of aging or senescence at the cellular level. And this was sort of the initial coining of the term senescence at the cellular level, or cellular senescence. So as these cells continue to divide, we now know, accumulate DNA damage and undergo a conserved cellular program called cellular senescence. A pre-senescent cell may have dysfunctional telomeres, and this is likely what was going on in Hayflick's experiments. With repeated divisions, telomeres shorten. This eventually leads to DNA damage and subsequently growth arrest, resistance to apoptosis, and profound alterations in gene expression. It's not just DNA damage that can trigger senescence. Other non-genotoxic stressors can also trigger senescence. What does this mean in terms of functional tissue? We have normal healthy tissue. We have some sort of injury. We know that sometimes with injury, cells are going to undergo apoptosis. We know that some cells may undergo frank DNA mutations. And then we have other cells still, that may undergo the senescence program. And this senescence program is not inert. The senescence program upregulates a wide range of secreted factors, and these secreted factors can influence neighboring cells. So this conserved cellular program exists to stop damaged and dysfunctional cells from proliferating. How can we tell that a cell is senescent? Well, there's a range of phenotypes. There are morphological phenotypes, and this is particularly useful in vitro preparations. There are increased expression of a range of senescence-associated genes, including P53, P16, and P21. All of these are going to be involved in the DNA damage response and then cell cycle arrest. In addition, senescent cells upregulate a senescence-associated anti-apoptotic pathway, as well as a range of what we call senescence-associated secretory programs, SASPs. And these factors that get secreted include a range of pro-inflammatory cytokines and chemokines, matrix metalloproteases, trophic factors. So a range of signals that can influence neighboring cells. Another marker would be the increased expression of a senescence-associated beta-galactosidase, and we'll actually use that as one of our markers. So as I mentioned, the senescent cells can upregulate a range of factors. This includes things like IL-6 and tumor necrosis factor alpha, as well as matrix metalloproteases, which can result in extracellular matrix remodeling, trophic factors like VEGF. Now, transient senescence is a normal part of development. It's a normal part of regeneration after injury, and it's a normal part of wound healing. So transient senescence is a normal part of our biology, and it is adaptive. However, in chronic cases and chronic conditions like cancer, aging, diabetes, tissue fibrosis, senescence may actually contribute to tissue pathology and ongoing damage. So again, in health, these cells are involved in tumor suppression, development, and wound healing. But in disease, they can contribute to a wide range of disease states, including cardiovascular disease, cancer, diabetes, neurodegenerative diseases, ocular diseases. And importantly, growing preclinical evidence demonstrates that removing senescent cells can improve outcomes in these models of chronic aging-related diseases, including osteoarthritis, chronic kidney disease, Alzheimer's disease. So what is our evidence that cellular senescence happens in the brain? First, it's been identified in the brains of individuals with Alzheimer's or ALS. This study by Turnquist et al. Showed quite robust increases in both p53 and p16 expression in brains from individuals who had died with Alzheimer's or ALS. As I mentioned, clearing senescent cells can normalize function in Alzheimer's disease mouse models. And p16 and p21 are increased both in Alzheimer's mouse models and in brains of patients with Alzheimer's disease. So in a range of disease states, increased senescence in the brain has been observed. But what about epilepsy? And why do we think that senescence may play a role? The thing that really got me started on this was the issue of DNA damage. And as I started thinking about the Venn diagram, the overlap between epilepsy and senescence, it got sort of longer and longer. So DNA damage, oxidative stress, pro-inflammatory cytokines, neurodegeneration, cognitive dysfunction, all of these are phenotypes that we see in our animal models of epilepsy and are observed to varying degrees in individuals living with epilepsy. So this led us to wonder, is there a role for senescent cells in epilepsy? Will an epileptogenic insult increase senescent cells? Are those senescence-associated secretory factors part of the chronic inflammatory state that's observed in epilepsy? And how might all of this contribute to hyperexcitability? So to begin to address this, we first examined the expression of p16 in the hippocampus of patients with temporal lobe epilepsy who had undergone surgical resection to treat their epilepsy. This is work that was published with Ty and Dave as co-first authors in Annals of Neurology earlier this year. And then this URL will take you to the paper if you're interested. So on the left, you can see the control case. On the right, the temporal lobe epilepsy case. You can see some brown dab staining indicating senescent cells, and you can see an increase in that p16 staining in the TLE condition. And in fact, we see about a doubling of the number of senescent cells in our temporal lobe epilepsy case as compared to our autopsy control cases. What was noteworthy about this was our autopsy control cases were actually about 20 years older than the temporal lobe epilepsy case. So this likely underestimates the overall magnitude. When we went back and examined the subset of patients that we had clean surgical outcome data on, we compared those individuals with Engel class 1 outcomes, so folks who were completely seizure-free, to individuals with Engel 2 or lower outcomes. And we found significantly higher senescence burden in the class 2 plus outcomes. While this was a sort of secondary analysis and not particularly well powered, it's sort of an intriguing observation. So great, we see an increase in senescent cells in the brains of individuals, in the hippocampus of individuals with temporal lobe epilepsy. What sort of cells are undergoing a senescence phenotype? To address this, we performed multiplex immunofluorescence, staining for markers of microglia, oligodendrocytes, astrocytes, and neurons, and then co-localizing with p16. The sort of punchline of this is we see an increase in senescence in microglia. We see an increase in senescence in oligos. We see an increase in senescence in astrocytes. And we see sort of a trend towards an increase in neurons. I'm not going to really come back to the neuron piece during this talk, but I have to say it's a piece that I find really intriguing because I don't fundamentally know what it means for a terminally differentiated cell to become senescent, and I'm very curious to explore further how p16 positive, putatively senescent or senescent-like neurons how their physiology may differ from non-P16 expressing neurons. So won't be able to show you any data on that, but that's one of the directions we plan to move over the next couple of years. So we have this robust elevation of P16 in glial cells. We next wanted to turn to a rodent model so we could start to address causality. The model we're using is pilocarpine-induced status epilepticus in mice. So we induce status with pilocarpine. Animals are allowed to have status for 2 hours, and then we stop the seizures with diazepam. And then we begin senotherapy. That senotherapy is going to continue throughout the latent period and through the period where we're going to perform EEG monitoring and cognitive testing. And this is going to be sort of the main model for this part. As I mentioned, one of the reasons that I was interested in senescence is one of the most potent drivers of cellular senescence is DNA damage, double-stranded breaks. And there's strong evidence that acutely after status epilepticus, there is a rapid induction of DNA damage. And in fact, when we stain for gamma H2AX, which is a histone phosphorylation marker that's a robust marker of DNA double-stranded breaks, you can see down here, this is 2 hours after status epilepticus, the hippocampus is just lit up with gamma H2AX. So we have this really robust induction of DNA damage with status. So I was pretty sure, I convinced myself that we were going to see a pretty rapid induction of cellular senescence. It turns out I was wrong. And our early time periods showed very little in terms of senescence markers. And in fact, I have to give credit to Ty for this. We were starting these studies during the pandemic, and we were doing all of these early time points, and we weren't seeing much. And she had a cage of animals that had sat for a while after status, and said, well, I think I'm just going to slice them and stain them. And I said, yeah, go ahead. You're probably not going to see anything. But sure enough, she did. So with time after status, we actually get this sort of nice upregulation of P16. And I'm showing you here transcript levels. This is in situ hybridization for P16 and P21. That's CDKNA2A and 1A. And you can see 2 and 8 weeks after status epilepticus, there's a robust increase in the number of cells that are expressing P16 or P21 mRNA in the hippocampus. The same thing is true when we look at protein, and we can do this in a couple of different ways. We have a reporter line that expresses the fluorescent protein TD tomato under the control of the P16 promoter. And you can see 2 and 8 weeks after status, an increase in TD tomato positive cells. You can see this with immunofluorescence for P21. And as I mentioned before, we can stain for the senescence-associated beta-galactosidase, and we can see an upregulation. You can see that this upregulation starts 1 day after status, but it's not all that robust. By 2 weeks after status, it's quite significant. And then it continues to increase in the weeks and months after status epilepticus. This effect is seen across all of the hippocampal subfields, shown quantified in black for the dentate, gold for CA3, and blue for CA1. So across all the subfields, we see the same time course of increase in P16 positive cells after status epilepticus. So I showed you in our human tissue that we had quite robust localization with microglia, astrocytes, and oligos. What about in our mouse model? So in our mouse model, we saw the most robust colocalization with microglia, followed by astrocytes and oligos. So this is our colocalization in panel B with microglia. Again, 2 and 8 weeks out, you can see a significant increase in colocalization of the transcripts for AIF1 and P16. And we can see this quantified over here for total number of microglia, which again, we expect to sort of increase over time after status. So, but we still are seeing, again, this co-localization with oligos, astrocytes, and microglia. But given that microglia showed the predominant phenotype, that's really what we focused on. One of the ways that we've addressed this is using a reporter line where we can cross that P16TD tomato reporter with a CX3CR1 EGFP line that labels microglia. And what this results in is some microglia that are, all microglia are green. Some microglia are also red if they are senescent. So in a field, we can see some microglia that are green and some that are green and red or yellow. And then we can quantify based on that the morphology of the microglia. So some confocal images of various microglial morphologies and using some automated approaches to quantify microglial morphology, we found a decreased cell territory volume in P16 positive microglia after status epilepticus, a decrease in average branch length, and a decrease in max branch length. All of this is consistent with them taking on a more amoeboid-like morphology. As they become, quote, activated and reactive, they undertake this more amoeboid morphology. So there's a change in morphology of the microglia that are expressing P16 after status epilepticus, but how about their function? So to do this, we're going to use a slice preparation. Microglia are continuously surveilling their environment. This is their normal homeostatic role. And in acute slices from hippocampus, we can do confocal live imaging and monitor how these microglia extend and retract their processes over time, how they surveil their environment. It's pretty well established that status epilepticus changes the motility of microglia. This is in the acute phase after status. There is an upregulation in motility of microglia, and you can see this time-lapsed here in the control. At the center here, there's a pipette that contains some ATP, and you can see with time, microglial processes migrate towards that ATP source. You can see both after electroconvulsive shock and after status epilepticus, there is an increase in the velocity by which they approach that ATP source. This is just an example showing some of the live imaging that we can do. And you can see this here as well. So here we're imaging microglia, and you can see here some microglia that are clearly P16 positive, some microglia that are not. So in this case, we measured microglia from animals that had status epilepticus and animals that didn't have status epilepticus, and we compared P16 negative to P16 positive microglia. Our overall finding in both the P16 positive and P16 in the P16 positive microglia from both the status and the no status conditions was they had decreased overall motility. So they moved around less, they surveilled less, suggesting that the senescence-like phenotype impairs some of their homeostatic functions. So just to quickly recap, I've shown you that senescent cells accumulate in the hippocampus of patients with TLE, that they accumulate in the hippocampus of mice after status epilepticus, that this is predominantly a glial phenotype, and that morphology and function is altered in these senescent glia, in particular microglia. So the next question is, what happens if we remove these senescent cells? We're going to do this in a couple ways. First, we're going to use a genetic strategy. So this is a mouse called the P16 Inc attack mouse. So this mouse, under the control of the P16 promoter, has an inducible cassette that expresses caspase 8 as well as an FKBP domain. So in order for caspase 8 to activate, we need to dimerize it. And we can do that with a drug that binds to this FKBP domain acts as a dimerizing agent, and this drug is, by the way, called AP, and allows activation of caspase 8. So we can give AP. It's not going to do anything to a cell that isn't without an active P16 promoter. But in a cell with a P16 promoter, we're going to get expression of this construct. We're going to get AP there. It's going to dimerize it. It's going to activate caspase 8, and that's going to trigger apoptosis and kill the senescent cells. So here's our timeline. We're going to give status epilepticus to 3-month-old mice. We're going to start treatment with AP right after status epilepticus. We're going to monitor them over time. We're going to perform a series of behavioral tests, followed by either EEG analysis, slice physiology, and then histology. So right off the bat, I can show you that P16 is decreased in animals treated with AP. So our genetic senolytic therapy reduces senescent cells, and it's about a 50% decrease. So here's the vehicle control, here's the AP condition in red, and again, about a 50% decrease. As I mentioned, we did a whole range of behavior. Our hypothesis was that SC ablation, senescent cell ablation, will normalize behavior after status. I'm only going to show you a few pieces. The first is the Barnes maze, so a test of hippocampal-dependent spatial memory. You can see in gray the no status epilepticus condition across training days. We're looking at escape latency on the Y. In black, the status epilepticus condition, and you can see that these animals basically fail to improve over training. And then in red are AP treated, so our senolytic treated, and you can see that we actually restore them back to control levels by the end of training. You can see that quantified here on the probe day. The latency to escape was around 50 seconds in the no status condition. It's around 80 seconds in the status plus vehicle condition, with many of the animals failing to escape, and then brought back to control levels by senotherapy. We also examined a novel location task. So 2 objects that are identical, we allow the animal to explore, we take them out, we move one of the objects. Rodents will normally have a preference towards the novel location. You can see that preference here, 50% or 0.5 would be equal preference, so chance. Our no status condition clearly prefer the novel location. Our status condition essentially at chance. Our status condition plus senotherapy back to control levels. In addition to behavioral testing, we also examined long-term potentiation in the hippocampus of animals after status treated with either vehicle or AP or senotherapy. You can see the average LTP in our vehicle-treated animals here, and you can see that following treatment with AP, the long-term potentiation was significantly increased. So all of that's well and good, but ultimately, if we're aiming to treat epilepsy, we care about seizure burden as well. So what happened when we monitored seizures? So we performed 10 days of video EEG monitoring. You can see here in our control condition, our vehicle condition, status plus vehicle, the animals are having seizures that are pretty typical for the model, between, 1 every couple of days up to several a day. In animals treated with AP, our senotherapy, we significantly reduced the average number of seizures the animals had, and we protected a large number of animals from spontaneous seizures at all. This resulted in an overall lower cumulative seizure duration without any change to the average duration of an individual seizure. So this genetic strategy is a useful experimental tool. It gives us some really good specificity. But if we want to do something translational, we need to think about pharmacological strategies. And fortunately, there's a range of compounds that have been developed, like dasatinib and quercetin, which are both targeting tyrosine kinases, and I'm going to show you data from these. And then other compounds like navitoclax, which targets the senescence-associated anti-apoptotic cascades through BCL2. And then there are some drugs. We're gonna use one called SSK1, which targets the senescence-associated beta-galactosidase. So we have a range of pharmacological targets, and we next wanted to see if using these targets could reduce senescent cells and improve epilepsy outcomes. Here's just a quick indication. As I mentioned, dasatinib and quercetin are both kinase inhibitors. And then we have prodrugs like SSK1, which can be converted by senescence-associated beta-galactosidase into a senolytic. So dasatinib and quercetin, I'll call them D and Q, are easily the most widely tested senotherapies across a range of indications. And they have been shown to reduce age-associated inflammation and cognitive dysfunction, aid beta plaque reduction, and rescue of cognitive function in a mouse model of Alzheimer's disease, reduce tangles in a tau model of Alzheimer's disease, and then anxiolytic effects in an obesity-induced anxiety model. So a pretty wide range of effects. SSK1, or selective senescence killing compound 1, is a clever compound. So this is a gemcitabine analog. It's a prodrug for gemcitabine, in fact, that has a domain that's hydrolyzable by the senescence-associated beta-galactosidase. As a result, you only get generation of gemcitabine and cytotoxic effects in senescent cells. And you can see cell viability here for SSK1 in non-senescent cells, 100% viability as you increase concentration of SSK1. Senescent cells, we get a concentration-dependent decrease. So both SSK1 and DQ reduce senescent cells after status epilepticus. Again, we get about a 50% reduction in senescent cell burden here with DQ, vehicle in black, DQ in blue. And then here for SSK1, our no status condition, our vehicle condition in red, and our SSK1 condition in blue. And the SSK1 data were just published in Epilepsia with Dave McFall as the first author. And again, this tiny URL will take you to the paper if you're interested. So same behavioral tasks as before. I'll just show you a little bit of data. DQ improved performance in the Barnes maze, just like genetic senotherapy did. Again, no SE. SE plus vehicle, basically at ceiling, and then SE plus DQ down here, basically back at control levels. Same thing in the novel location task. Significant preference for the novel location in our control animals at chance after status epilepticus, normalized by DQ treatment. Same story with SSK1. We normalize learning in the Barnes maze with SSK1 in blue, status in red, and control in black. So SSK1 is sort of here in the middle. And then basically normalize preference in the novel object location task as well. And both SSK1 and DQ protect against seizures. So you can see again here, the average number of seizures per day are significantly reduced in animals treated with DQ, as is the cumulative seizure duration and the average seizure duration in this case, which was sort of interesting. With SSK1, these effects are even more striking. This is the total number of seizures over the monitoring period in vehicle-treated animals versus SSK1-treated animals. So this would work out to about 2 seizures a day. But you can notice here, there's a large number of SSK1-treated animals, in fact, 12 out of the 20, that did not develop any spontaneous seizures after status. So we protected a significant proportion of the animals from developing epilepsy by applying SSK1 senolysis immediately following status through the end of the experiment. So I've shown you that senolytic therapy normalizes behavior. This is spatial memory after status epilepticus. It reduces seizure burden and disease penetrance after status epilepticus. So what about other seizures or epilepsy types? So I want to tell you about some work we've been doing testing the efficacy of senolytic drugs in post-traumatic epilepsy. And I want to just first say why post-traumatic epilepsy. It's got a fundamentally different etiology, right? I mean, this is an acquired injury, and it is a significant cause of epilepsy. And there's a certain translational appeal for post-traumatic epilepsy. With post-traumatic epilepsy, we know when the injury happens in a patient. For individuals that develop temporal lobe epilepsy, sure, if they have complex febrile seizures, they're at a heightened risk for later life TLE, but it's hard to imagine treating individuals from, early life for decades. With PTE, if they have a penetrating trauma and an early post-injury seizure, I mean, there's a reasonably high likelihood that they'll be developing epilepsy within the next year. So it makes it a much more tractable translational angle. The model that we're using is the controlled cortical impact model. We're using a 2 millimeter CCI in mice. This has previously been shown to trigger both senescence and seizures in separate studies. Our treatment is going to be D&Q or vehicle. Just as in the last study, we're going to do some behavior and then some EEG monitoring. First, we're showing an increase in senescent cells following injury. And you'll note that this is a little bit more rapid than it was in the status epilepticus model. As soon as a week after injury, we're starting to get increases in senescent cells. And again, these are co-localized with microglia in the hippocampus. You can see some in the hilus here, nicely co-localized between EBA1 in green and P16 in red. And they're also co-localized with astrocytes. So this is co-localization, top row DAPI, second row GFAP to mark astrocytes, third row P16 in green. And again, you can see increased co-localization between P16 and, well, first this is showing total P16 cells, but as well in a significant co-localization with astrocytes. So senolysis modestly improves learning and memory in these animals, and the effect really is modest. So in red, we have our CCI condition treated with vehicle. In blue, we have our CCI condition treated with D&Q. While there is a significant improvement in learning, there's still a far way off from the control condition. What about seizures? So I have to say this was disappointing. Senolysis does not protect against PTE following severe injury. You can see this is pretty well powered, and there's really no difference in either the number of seizures nor the penetrance of injury of seizures after brain injury. So about 70% of control animals, vehicle-treated animals, and about 70% of DQ animals had spontaneous seizures. So why the difference from our TLE model? Well, one possibility is we use a quite severe injury here. With the 2 millimeter CCI, there's very little ipsilateral hippocampus left as we deploy it. So that's one possibility. And we're actually going to be repeating these experiments with a milder injury. The issue with a milder injury is the fraction of animals that develop spontaneous seizures is significantly lower, so it requires larger samples to be able to detect a treatment effect. The other piece may be timing, and we actually have an experiment going on right now comparing immediate senolysis, so starting treatment right after CCI versus delaying by 2 weeks to allow some of the normal post-injury healing to occur more. So more to come on the CCI story, but we're getting close to getting the data together. So what about other seizure and epilepsy types. I want to tell you a little bit about some of our data in a mouse model of Alzheimer's with comorbid seizures. And first, why? Again, this is a different etiology. This is a genetic model of Alzheimer's disease. These are the APDE9 mice. These are presenilin 1 and APP mutant mice. They develop predictable seizures after 4 or 5 months, and those seizures increase in frequency as the animals age. This is a patient population where senolysis is already in some clinical trials, and it's an animal model where senolysis has already been shown to improve some of the learning and memory deficits, but nobody's previously looked at the seizure phenotype, so that's what we sought out to do. As I said, these animals have the Swedish mutation in APP as well as an exon 9 deletion of presenilin 1. About 50% of the animals have seizures by 4 months, and DQ treatment reduces plaque burden and improves cognitive impairments in these mice. So first off, early DQ treatment reduces both senescent cells and A-beta plaques. You can see this here. In the vehicle-treated animals, there's a significant increase in the number of P16 cells. This, in fact, is brought down to control levels by DQ treatment. And same thing is observed when we use senescence-associated beta-gal as our marker. You can also see a reduction in A-beta plaques following DQ treatment. So interestingly, early DQ treatment also reduces seizure burden. And again, this is at 4.5 months in 4.5-month-old animals. And you can see, again, a significant reduction in the number of seizures, the seizure duration, and the percent that were seizure-free at 4.5 months following DQ treatment. So what about late treatment? We're not likely to start somebody on senotherapy early in life with the goal of preventing later Alzheimer's-associated seizures. So what about later treatment? So we reduce senescent cells. So that's on the left here. Senescence-associated beta-gal is down in the APDE9 mice treated with DQ, but not plaques. So what about learning and memory? So it still improves learning and memory. So even though we're not reducing plaques, we are reducing senescent cells, and we are basically returning learning and memory to the levels of wild-type age-matched controls. So what about seizures? And I should mention that the behavioral testing was done at 14 months here. So this is 16 months when we're doing the seizure monitoring. It actually made the seizures worse. So when we quantified the total number of seizures, the vehicle treated, there was a significant increase following DQ treatment. So we have a little bit of a paradoxical effect here. Early senolysis protects against seizures in an AD model, but late worsens it. And what we would really like to understand is what is it about the timing that leads to this difference? So in the last minute or two, I want to tell you about one last aspect of senescence and epilepsy, which is how senescence and senolysis impact seizures in normally aging animals. So late onset epilepsy is of increasing appreciation and concern. It is really well established that the aged brain has an increased vulnerability to seizures. And we already know that aging accumulates senescent cells. So our question was, does senolysis protect against age-related vulnerability to status epilepticus? Whoops, sorry. So here's our paradigm. We're going to take 14-month-old mice. We're going to treat them with D&Q for 16 weeks. We're going to test them on the Barnes maze to see if we've improved any cognitive function. We're going to induce status epilepticus. We're going to monitor the status and the survival, and then we'll do a water maze test at the end. And I should say, we published this a couple years back in Aging Cell, and again, link to the paper down there. So I'm showing you here Kaplan-Meier plots for the probability of status epilepticus with time after pilocarpine administration. The first thing I want to point out is in the young animals here, these are 3 to 4 month old animals. The dose of pilocarpine we're using is resulting in status in about 40% of the animals. This was by design because in our aged animals, you can see that here in red, 100% of the animals develop status, and they develop it quite rapidly. So this is what I mean by an age-associated increase in vulnerability to status epilepticus. It's that difference between this blue and red curve. So a DQ treatment reduced the proportion of animals developing status, and it delayed the onset of status significantly. It also significantly increased the probability of survival in these animals. So we had about 50% mortality in the aged vehicle animals, about 10% in the controls, and about 40% in the that's backwards. About 40% in the young animals and about 10% in the controls. Animals that were treated with DQ, aged animals treated with DQ, displayed less severe seizures during status. They had a longer latency to status. And unsurprisingly, again, they had a reduction in the number of senescent cells. So it's not just my group who's interested in senescence and epilepsy. There are a couple of others that are doing some really interesting work. This study has demonstrated increased P16 and P53 expression in tissue resected from patients with type 2 focal cortical dysplasia, and they demonstrated that DQ reduced seizures in an mTOR mutant transgenic mouse. This study shows at the transcript level senescent cell markers that are overexpressed, particularly in individuals with drug-resistant epilepsies. And this is a really interesting angle as well. Not only could senescence contribute to epilepsy, as I think I've shown you today, but maybe it also plays a role in the drug resistance. And this is an angle that we're also keen to explore. So I think that there's a lot of future work to be done. As I mentioned, while dasatinib and quercetin are one of the most commonly used senolytic cocktails, and it is in clinical trials for several age-related diseases, it is not approved as a senotherapy. And we are just beginning to understand some of the side effects that may be associated with senotherapy. So this was a paper that was published just earlier this year showing that senolytic treatment induces oligodendrocyte dysfunction and demyelination in the corpus callosum of normal young animals. So the quest for sort of more targeted senotherapies is a high priority, and understanding optimal timing is also a goal. So we want to work towards defining effective windows for intervention and probe deeper the mechanisms by which senescent cells lead to seizures, and fully understand this interaction between injury type, severity, and timing of treatment. So I want to come back to sort of this cascade again. We have epileptogenic insults and then a cascade that leads to epilepsy. During this latent period, what we think might be a paradigm shift are the identification of disease-modifying therapies. And we propose that senolysis may be a disease-modifying therapy in this case. The other piece, and this is, again, one of those questions of timing, what happens if we do the senolysis late? What happens if we wait until after the onset of spontaneous seizures and then remove senescent cells? Will we reduce the seizures? Will we change the disease progression? Again, a high priority for us to examine further. So with that, I'm happy to answer any questions. Thank you all for listening, and thank you, Louisa, for the opportunity to speak with you all.
Thank you so much, Patrick. I appreciate a lot. And I want to know if there are some questions. Javier, for example, do you have any question?
Yes, I have. Hi, Patrick. Thank you. Thank you very much. Super nice talk and super interesting. We work in glia, so we are really happy of having people talking about epilepsy and glia together at the same time.
Absolutely.
Yeah. So my question is, what's going on with this microglia that is senescent?
Yeah.
Are they doing less pruning, so you have sporiosynaptogenesis everywhere, and they are not working properly? I don't know. That's my first hypothesis, but for sure you will have some.
Yeah, so I think it's a great question. So we know what we know right now is they have decreased motility, which would certainly be consistent with less of the homeostatic sort of pruning functions. We haven't looked at pruning, so I can't comment on it specifically, but that would absolutely be consistent with that. The second piece is we know that senotherapy, and I didn't show these data, can normalize some of the pro-inflammatory cytokines after status epilepticus. So it may be that these senescent microglia are some of the sources of those pro-inflammatory cytokines. We have a set of experiments, and I'm waiting on the sequencing data right now. We've done some RNA-seq to try to understand specifically altered gene expression in senescent microglia after epileptogenic insults. And I think that that'll give us some indication. The other thing that we're hoping to be able to do, we now have a genetic model where we can selectively get rid of just the senescent microglia or just the senescent astrocytes or just the senescent whatever, right? So, and this grant is going to get reviewed this summer, so keep your fingers crossed. As I'm sure you're all aware, funding right now in the United States is an absolute disaster. So,
Yeah.
but one of the things we're proposing to do is cell type specific senolysis, because that'll actually tell us if it's just the senescent microglia. If we get rid of them, we should recapitulate the same effects we get with systemic senolysis, and that'll let us dive deeper. So a lot unanswered, and I can't really give you more than that yet because we haven't done the experiments.
Yeah, perfect. And my other question was, at the beginning, I say, okay, astrocytes are senescent, so they are not providing, lactate and everything to neurons, and they are not, shutting down the reactive oxygen species and blah, blah, blah. But no, the effect that you saw is more, it's more in microglia compared with astrocyte. True? Is that true?
So there are more senescent microglia than there are senescent astrocytes in the mouse model, but there is also a significant population of senescent astrocytes, and we haven't even touched that. Again, in the grant, we propose to do selective ablation of those senescent astrocytes to see if they contribute. And there's actually some really interesting data from Chris Dula's lab at Tufts, where they've described these senescent-like astrocytes in aging and some altered function in them. So I'm sort of curious about exploring that in our context as well. We just haven't gotten around to it, and it's definitely a priority.
Okay. Thank you. Thank you so much. Thank you very much.
My
Thank
pleasure.
you. I want a question, Patrick. You analyzed brain tissue obtained from patients with drug-resistant seizures.
Yep.
And also, you analyzed the brains of animals submitted to status epilepticus. In both, there are significant changes. Yes?
Yes.
In this case, senescence is a biomarker of epilepsy or only drug-resistant epilepsy? Because pilocarpine seizures induce both epilepsy and drug-resistant epilepsy. What do you think?
So it's a good question. I think that it may be a more general biomarker or contributor only because we also see this accumulation in our CCI model as well as in the Alzheimer's model. But in the pilocarpine model, I think that this is one of those areas that we're proposing to try to examine in the next grant, right? The interaction between senescence and pharmacotherapy. Because we could segregate out those animals into the drug resistant versus the non-drug resistant. And the hypothesis would be, based on some of the human data, that we would see higher senescence burden in the drug resistant animals. We just haven't tested it yet.
Okay.
Yeah.
Probably just an acute seizure can produce some senescence.
So I'm trying to think if we've looked at an acute seizure. We have not looked at a single acute seizure, aside from status. But we have also looked at this and I mean, these data aren't published, and they weren't fully powered, but the patterns were the same. We saw this in the rat kainate model, and we also saw this with amygdala kindling. So it's not just seizures that are kind of damaging seizures, because like the kindled seizures, I mean, you're not getting massive damage after those seizures. And even after early stages of kindling, we can start to see an increase. So it would be sort of consistent with even kind of brief activity may be sufficient to start to drive senescence. And then one of the questions is, does it increase because the emergence of spontaneous seizures then make more cells become senescent over time? And we really don't know cause and effect there yet.
Okay. And other question is about post-traumatic epilepsy. You apply the elimination of senescence after the TBI.
Yes.
Yes?
Yes.
But how about as a preventive therapy? Is it possible?
It is possible. The challenge is we've done it thus far in relatively young animals, so they don't have much of a senescence burden in the absence of some sort of injury. So we could definitely give it a shot. And again, given I'm also sort of interested in these late onset epilepsies, and certainly the older animals are more likely to develop PTE. So, you know, very similar to how we did with the status experiments, we could, you know, age animals use a milder injury. We would expect a higher penetrance of PTE, and then compare senotherapy. That would be sort of how we'd have to do that experiment.
Okay. There is a question from a lady, Patricio, that she's a scientist in Puebla, very close to Mexico City. Have you been able to dissect whether the anti-epileptogenic effects of senolysis are primarily driven by, let me see.
I got it here.
By the clearance of senescence microglia versus astrocytes, and whether this correlates with specific changes in the CSHP profile.
Or other factors that are directly modulating excitability. Yeah. So excellent question. We don't know yet. I think that it is unlikely to be, this is just my hunch, but given the fact that folks have tried a lot of interventions targeting various pro-inflammatory cytokines sort of one at a time, and even sort of broader anti-inflammatory therapies, and they've not really had robust anti-epileptogenic effects. My hunch is that it's not just the SASP profile, that it's going to be a combination of the SASPs, which I am sure are contributors, effects on synaptic excitability, and then effects on the homeostatic functions of the glial cells. The last piece is, like, as I mentioned early on, we also have neurons that are expressing P16, and I don't know yet. You know, one of the things we want to do is we want to patch the P16 positive versus P16 negative neurons, and see if the physiology differs, because there may be intrinsic excitability differences as well in a neuron following induction of this cascade. And we just don't know yet. NIH needs to fund my grant.
I have another question. You presented some side effects induced by some drugs inducing the elimination of senescence.
Yeah.
Those side effects are induced by the drugs or because the elimination of the senescence per se?
Good question. In that case, it's probably by the drugs. So dasatinib is it's a tyrosine kinase inhibitor. It hits like AKT, ABL, a whole bunch of tyrosine kinases. It is not a particularly clean drug, and it's not surprising that there are going to be sort of side effects from that, right? That said, getting rid of senescence can also do things like impair wound healing. So one of the things that we have to be careful about is, we have to implant these animals for EEG pedestals. And in the PTE case, we actually stop DQ right at the time that we do the surgery for the EEG pedestal, because we're worried about survival and healing after surgery. So there are definitely going to be side effects that are senolysis mediated. And then given that these are relatively dirty drugs that we have so far, there are side effects that are likely to be off-target effects. One of the ways we're trying to get at that is we have our genetic method, we have DQ, we have SSK1, and then actually one of my postdocs is running a study right now with navitoclax, which is a BCL2 inhibitor. So where we can see convergence on side effects across those mechanisms, that would strongly support that there's senescence associated. And where we can see sort of different off-target profiles, that sort of, it would indicate to us that it's, mechanism specific.
Okay, my last question. How about optogenetics to eliminate senescence?
I don't know how we would do it with optogenetics, but what I would like to do is I'm going to answer a different question than the one you asked. What I would like to do is I would actually like to activate those senescent neurons, just those senescent neurons. That I can do. That we have the genetics that we can do, and see if either turning them on or off, not sort of optogenetically eliminating them, but optogenetically turning them on or off, if that would change the seizure phenotype. So that's something that we might give a try to. We have to do a few more mouse line crosses to be able to get the right combination that we need, but it's tractable, it's doable.
Thank you. Is there any other question?
There is a question in
Yes.
YouTube, because Patrick, we're streaming this directly to YouTube, and there were like 15 people more. So, but one of them, I think is Ana Montesinos. She did a question, did you observe any rebound or compensatory biological response after senescent cell clearance, such as accelerated reaccumulation of P16 positive cells or secondary inflammatory activation?
Excellent question. So we've not done the experiment yet, but it's ongoing. If we give a narrow kind of burst of senotherapy and then discontinue it, will we get a rebound accumulation of senescent cells? We don't know. This is particularly interesting in the TBI case, because one of the things that we see in the CCI model is there is a really rapid induction of microglial number, just total microglial number. They proliferate like crazy after CCI. So one of the possibilities is that the senescence that happens is actually a replicative senescence in that case. So you would imagine that if we either blocked the microglial proliferation, we might actually be able to block the senescent cell accumulation. And alternatively, if we clear senescent cells, we might not actually see that rebound, because the cells are going to sort of gradually recolonize from the non-replicatively senescent remaining microglia. So it's an experiment that my postdoc Max is working on right now. Great question.
Thank you. Thank you very much.
Thank you.
Awesome.
No, probably no more questions. We really appreciate your conference in LabEpi. LabEpi, it's a network of scientists and professionals involved in epilepsy research all through Latin America. And we are so happy to have your conference, and I hope in the future you can visit one country of Latin America.
Absolutely.
Or all countries of Latin America.
I would love to. And thank you again
you.
for the invitation and for all the great questions. This was really a pleasure.
Thank you so much.
Thanks so much. Enjoy the
Very
rest
much,
of the day.
Patrick. Thank you.
Thank you.
too.
Bye.
Bye.
Bye-bye.